Hi, I'm Alexander Pelly. I'm one of the animal care technicians over in LSA.
What is the research name of Purdue University? Who do we fall under?
This is called EVPRP, which stands for Executive Vice President of Research and Partnerships. And that umbrella term refers to any of the animal-related research that goes on here at Purdue.
That's right. How many of each of the following animals are in the operation? How many mice do you think we have?
We, right now, have around 4,000 to 4,500 individual cages of mice. Mice are counted per cage, so there's anywhere between 1 and 5 mice in each of those cages. And we have room to grow.
How many rats?
Rats, right now, those are counted individually, not by cage, unlike the mice. We have 80 so far, ballpark number, and we also have room to grow for them as well.
How many other rodents do we have, or what types of other rodents?
So in terms of other rodents, you can find people using chinchillas, guinea pigs for research, and then not necessarily a rodent, but also rabbits are a big part of research at Purdue.
What other species are across campus?
And the different species across campus are very varied. Some of the more common species you'll see across campus are horses, cows, pigs, llamas, sheep, goats, fish, poultry, salamanders and frogs, and even turtles.
What preparation, education, and experiences did you obtain, does one need, or should one have to be successful in this type of industry?
So there's a lot of ways to go about it. Some people start from the ground up, start from scratch, and they come in an entry-level position and learn on the job. Part of that includes gaining certifications through our regulatory body, ANSC. Those actual certifications have 3 levels: an associate's, a technical, and then a technological level. And animal care technicians can apply to take those tests in order to earn their certifications. Also, a lot of us do have a bachelor's. For instance, I have a bachelor's of zoology, but anything biology or animal related would be useful in obtaining a job and working in a place that does animal research like this. And when it comes to management staff, they have an entirely other set of qualifications as well. Is that right, Mary?
Yeah, that's right. I'm Mary Larimore. I'm the manager for nutrition science and animal science. And we, all of the managers have bachelor's degrees, some higher. Mine is in animal science from Purdue. Whoop, whoop. And so, and we, some of us go to extra classes, like we went to llama, which is down in Clearwater, Florida. You go down for maybe 3 days, and it's just all the managers getting together and talking and troubleshooting. And then we also had ILAM, which was a 2-year program that we went to Memphis for a whole week and learned and studied together. So.
And regardless of your level here working at Purdue, there is a requirement for furthered learning, which is mostly done online through different kinds of coursework, through ALAS and also through LinkedIn, I think.
Yep.
Now it's LinkedIn.
Yeah, LinkedIn.
And
Lynda.com.
along with that, though, we also do and are lucky enough to have a training coordinator who trains all the research-related staff here on campus, and any of us are able to go and take the physical courses that have to do with technical procedures here in a lab space.
What are 2 major challenges facing your operation or industry today?
So the most obvious one that I can probably think of is grants, how the funding happens. Like I said earlier, it's up to the research staff to write their grants, get them submitted, and there is, of course, a department here on campus that helps all the research staff make that happen and continue to get funding. And on our side of it, we're a hard money department, so our budget is clear cut because we aren't the research staff, we're the husbandry side of things. and that work has to stay consistent in order for everything to get done right. So reductions of the researchers themselves, though, does affect our overall census, the actual population of animals we have, which does affect how much we're able to make through per diem costs. The other thing that's a little more esoteric is just public perception of research, specifically animal research. That is something that you may not have on a day-to-day situation, like you don't have to, fight people off of you when they find out that you work at a research situation. But it is something that you have to keep in the back of your mind and make sure that you really do have that ethic of confidentiality and reasonability to know what you're saying and when you're saying it. Because there is some negative public perception about the kind of animal work that can happen for further research into biomedical or agricultural or behavioral research.
And it's true, a lot of it is just perception. It's not reality.
No, and that's something that is a challenge for the department and really all across the nation, is making sure that the public has enough information to make an informed decision, but most of the public just doesn't have that base knowledge, which is when we talk about qualifications for working here. That's kind of where you get a lot of that base knowledge for what goes on in a research facility. But it is something that, hopefully videos like these help people understand what goes on a little bit better.
That's right. So what kind of job opportunities do you have here?
So in our department, from the ground up, I could say, you'll have part-time student care techs. So they're animal care techs for sure. They work part-time and with their schoolwork. And that's a great way to get hands-on experience working in the laboratory environment and really learning about the different kind of studies that can happen. Next step up would be any of those care tech levels I was talking about, from associate to technologist, where you're just a full-time animal care tech. Much more responsibility, of course, with that position. You're really managing those micro and macro environments to make sure that those researchers have consistency in their studies. Next level, of course, is the manager, like Mary here, right? They manage not just the facilities as they relate to specific departments like animal science or nutrition, but they also manage the actual care techs to make sure that everybody has the supplies and experience they need to make informed day-to-day decisions, and also to respond to situations that occur for health or other animal-related emergencies that can sometimes happen in a research environment. After that, both of our boss, the assistant director, she manages all of the centrally managed facilities here at Purdue, and her job is to make sure there's consistency and continuity throughout all of the facilities in how things are being done, so that if a researcher is working in one building, they can go to the next building over and they don't have to worry about any nefarious variables messing with their study. After her is the director, right? The director is in charge of
everything.
Everything.
Yeah, all the animal facilities and anything that goes on with the research,
so
everything.
They're working not just with our staff as animal care, in an animal care capacity, they're also working with the research staff on formulating how they're going about their studies. And they're also working with the veterinary staff to make sure that health concerns are followed up and taken care of efficiently. And then of course, there are vets, there's vets here that manage how responses are taken to health emergencies. And if the research requires it, how to manage certain conditions that are a result of research to minimize discomfort and distress on the part of the animals. So any of those different levels are job opportunities here at Purdue. And this is also from our perspective as the centrally managed facilities. Underneath the director, there's also uncentralized animal facilities that also do work at various satellite facilities and on campus as well, working with any number of species that were in that list previously.
That's right. So what recommendations do you have for me if I want to work in this type of operation in this industry?
So bottom line is experience counts. The degree counts, but for entry-level positions, experience very much counts. Like I said, here at Purdue, we have the part-time student options. That's a great way to get your feet wet and really get to know what's going on here before jumping into anything full-time. Also, a background in animal work or any kind of biological field really does help break a lot of barriers in actually understanding what the researchers are doing and the applications and the impact that those studies are going to have.
Yeah, you need to be able to look at an animal and see, is it in pain? Does it feel good? There's a lot of things that go into that. And so just animal work is important.
Yeah, the actual hours you're putting in. This is a typical mouse housing room. You see we have all these different cages here. Generally, for consistency, of course, in a room you'll have pretty much the same kind of caging. Each of these are micro-isolator ventilated cages, which means that these micro-environments contain everything the mouse needs, including air transfer, so that they're taking out old air that has ammonia from them urinating and puts in fresh air that is filtered with these blowers up here. Each of these cages can house anywhere between 1 and 5 mice. When it comes to breeder pairs, which I don't think we have in this room, though, you can have 2 mice, a male and a female, and their pups. And litters can be anywhere between 2 and 13, as far as most of the mice go. That's strain dependent, though. These setups are convenient for consistency with the lab work, but it also helps to really fine-tune any little changes that need to happen in the day-to-day care of the animals, if that is a study-related requirement, like for some of the behavioral research, for example. With these cagings, in this room, the bedding is really finely ground aspen chips, but across campus you'll see this, you'll see regular aspen chips. You can see, most commonly it's called betta cob, it's actually ground up corn cob. Great way to recycle that material. And it's a good substrate for the animals to nest in, and then also keep them warm, especially when they're nesting with pups. Enrichment is essential for healthy minds of any species, really. It's a standard enrichment here at Purdue to use this white crinkle paper. It's supposed to mimic their natural foraging behaviors and nest building because it's basically like grass. They have to pull it apart of the puck, which takes them a good long time, and that's something for them to do. And then they are very particular about where they like to put their nests and how they like to arrange them. It's very nice to watch them do that. And that's something that is required in order to make sure that these animals have a reasonable outlet for their natural behaviors. Because if you're not having natural behaviors, you're not going to get natural results in your study. So when you're working in the facility, right, you're looking at the cages. If you are a research investigator with these cages, or if you're an animal care tech, in order to monitor the cages, you pull them out. You'll take a look at the animals for their health, looking at their coat, looking at their, any abnormalities on their body. Some of it can be study specific, and it's important that we understand what's going on in the study to make sure that we can care for them appropriately. But if any work needs to be done with the animals, whether it's a procedure for the research or if the cages need changed by us for husbandry purposes, you'll have to make sure that you open them under a transfer station. So the transfer stations in here are these lovely phantoms.
Can you turn
real
them
simple.
on? Do you know how to turn them on?
Yeah, real simple. It's cool. They got a little touch screen on it. It has the lights, so it's perfectly well lit so that when you're doing procedures, you're not going in blind. And then it sets up an actual air current so that anything that is escaped from the cage while the cages are open underneath the hood, it's contained in that pocket of air under here. And when you're working, you'll want to make sure that you don't cover these vents here because that will disrupt the airflow. It's really important, even if you're not working with something that's, obviously a hazard, which can happen, of course, in research. Any of these animals will produce dander. You'll see allergens in their saliva or urine. Even things like these aspen chips can cause an allergic reaction. Any of the PPE we wear or these transfer stations are used so that we minimize the actual health risk for ourselves. When it comes to the health risks of the animals, it's also important. That has to do with things like making sure we have a mask on if we're working with immunodeficient mice. If we're working with the mice under the hood, it also makes sure that whatever air is getting into the cage while we're working with them, it's not coming from anything nefarious in the environment, in the macro environment of the actual room. Which, you wonder, what could that mean? Well, it's not always one investigator per room. A lot of the rooms here on campus, they don't need a huge colony. There is this concept of minimizing the actual number of animals you need to make sure that your actual research is done properly, but you don't need more research, or you don't need more animals to make sure that happens. But with that said, so that we can meet our per diems, we assign racks to multiple people. So if one researcher is working with a hazard and the other is not, you want to make sure that you are protected and that your animals are protected from that other hazard from the other study. That's why our protocols are very strict in terms of where you handle the animals and how that's done.
Hi, I'm Justin Murray. I'm an ALAT technician here. This is Alex. We already know him already. So Alex, what are the 3 R's?
So when you're going about making a new study, you have to justify what you're doing. Part of what I was talking earlier is that you don't need as many animals to get what you need done. So the 3 R's are refinement, reduction, and replacement. Not necessarily in that order. First thing you want to look at is if you can replace an animal model with a lower sentient organism, like a cell culture, for example. A lot of these studies will start using cell cultures before anything else, just to make sure if it's a drug discovery type thing, that those drugs are actually going to interact with their targeted cells in the way that they're assuming that they're going to through their preliminary work, just as an example. Refinement is, well, we already talked about reduction, which is you just need to know what is the minimum number of animals you need to get what you need done. And a lot of that goes into the initial work where you're looking at your studies that influence your own studies. So you don't just make something out of scratch. You have to go back in the literature and look at what other people have done in relation to the work that you're trying to accomplish and work with those numbers. Yeah. So refinement has everything to do with the technical applications of the procedures you're doing for your experimentation. There are rules associated with this sort of work, but in a general sense, working on the actual techniques for a specific procedure, if it reduces the amount of lives needed for that procedure, would overall reduce the number of animals you need eventually for the entirety of your study. There are limitations to that in terms of actual surgeries. Big invasive surgeries, of course, you don't want to put any animal through that many. So the limit for many of the large surgeries is just 1. But there's plenty of other techniques that have to do with blood draws or any other DNA sampling or imaging that have been and are continuing to be refined that help reduce the number of animals that you actually need to get your expected outcomes for your research.
So we're here in the rack room. Alex, you want to tell us about this lovely place?
Yeah, definitely. So we got a couple rat cages here. Again, like the mouse setup, these guys are on ventilated racks. A little bit different in their cage change schedule. We talk about budgeting out the time. They're still ventilated racks, but all these cages are a once a week change. Rats are a lot bigger than mice. They make a little bit bigger of a mess. The setup for these cages is a lot similar though. It's pretty similar though. Difference being that instead of one water bottle, they have 2 per cage. It's kind of nice that they're front loaded too, so that you can pop them in and out as you need to change them. And in terms of like the amount of mice or amount of rats, right, we don't count it by cage, we count it by individual rats. And the cage density requirements, you know, with mice you get 1 to 5, not including pups if you have a pair. With rats, it's actually a weight designation, so you can't have more than 900 grams of rat in a cage, which means if you have 300-gram rats, you're good. If you have 2 400-gram rats, you're good, like a breeding pair, for example. But just because it's a different setup in terms of how we do the per diem cost and the actual husbandry. These guys, hopefully you know, rats are very, very smart. They have enrichment just like the mice, who are also pretty smart. All of them will get an 8-gram puck. For singly housed though, since they're a social animal, we give them a little bit of extra enrichment in the form of these big red plastic huts, which looks like they kind of put them where they want them in order to nest, where they prefer, up in the back where it's nice and cozy and dark. So like I said, these guys are a once-a-week change because they make a bigger of a mess. The benefit of the ventilated racks for the mice and rats is that there's that air exchange going on. They're ventilated, so you don't have to worry about the ammonia buildup that you would get from constant urination that the animals would produce. But there are...
Does that come in with a static cage?
Well, I was going to say, over in his section, you'll see a lot of static housing as well. And then there's other various housings too, like maternity cages or what have you, where you would have to change it once a week because you don't have that ventilation. You don't want that ammonia to build up in the cage. It can become toxic to the animals and it's not a healthy living environment. So yeah, static cages, definitely a once a week change as well. And it has everything to do with making sure that the animals have a clean living environment. If the animals are healthy, you're going to get good data. If they're not healthy, your data could be for any number of reasons besides what you're trying to target information-wise as part of the actual procedures you're performing. Same setup in terms of like how you access the cages and do your procedures though. You want to get in your PPE, you're doing your health assessments, looking around to the cages, looking at their physical condition and everything. If you're doing a procedure or doing cage change, you're going to use the transfer station. It's another phantom, what are the odds? And same idea, right? You turn it on, you have to make sure that these air currents, I worked with these before, the air currents before you start using it are established so that this is all contained. There. Nice and well lit so that there's no guesswork in terms of what you're poking at or working with. Rats are a lot bigger, they're a lot stronger of an animal. So having your procedure, having your procedures done in a hood like this is helpful to remain in control while you're anesthetizing or doing injections or anything like that. And any of these hoods have the adjustments on them. Because when you're working, just an ergonomic thing, right? You want to make sure that you're not breaking your back and that you're comfortable working. Because if you're comfortable working, then the animals are going to have an easier time of it.
What are some of these rats used for?
So, a lot of these rats are for specific studies. Some of them are actually teaching rats, though. And by teaching rats, or teaching animal in general, through veterinary courses here at Purdue, the animals are used to demonstrate and have students learn certain procedures for veterinary application. When it comes to the studies, any number of things are used with rats and mice. They're very good models for disease, infection, and behavioral monitoring type stuff. One of the studies here, for example, works on fracture studies and how that process happens naturally, and then ways to have an intervention in those processes and kind of expedite the healing process, which is really fascinating. And I know some of these rats historically have gone through studies related to that to learn how that process happens and learn how we can expedite the healing process. any of the animals that you see on campus that are used in research, they all have very refined purposes, whether it's teaching or study related. And when we talk about the 3 R's about refinement and reduction, you know, there's the whole hierarchy of sentience in terms of what you want to start at and what you have to start at in order to progress your research. So things like that fracture study, you know, in terms of the actual intervention through drugs, those had to be tested on cell cultures first before they were even put in an animal model. Then the next animal model up potentially could be mice, and then after that something a little bit bigger like a rat, and then eventually something even bigger like a dog would be a model that would more closely reflect how those kinds of drugs would work in a human body, because, you know, they're heavier like we are. A rat body isn't necessarily getting the same load in terms of like a broken bone that a dog or a human would have. But it's important to have that process of starting small in order to make sure that you're not sort of, you know, going in blind to something that you're not prepared for. But it even comes down to surprisingly similar physiological things with other species. One of the main species used in Alzheimer's research, for example, are zebrafish. And a lot of amphibians are like this too, not necessarily for Alzheimer's, but it's because of their regenerative properties, something that humans don't have. But because of their regenerative properties, we can study similar pathways in their brains to influence how we go about designing drugs and rehab type stuff for our own purposes, for our species. But anything under the sun is possible. Just to reiterate that you don't just get approved, even as cool as it sounds, whatever your research is, oh, we're going to heal all the broken bones, or oh, we're going to cure Alzheimer's, or oh, we're going to find out the root of autism, things like that. You have to go through the proper channels. You get it approved through the PAYACOOK, the Purdue Care and Use Committee. You have to make sure that the veterinary staff have their input on terms of interventions, the drugs used for anesthetizing or otherwise, and then you have to make sure that that's communicated to the care staff, our job, so that we know what to do for the animals in terms of their husbandry. All that stuff is so important for your actual funding as you go about defining what your research is. When you look at a study like a fracture study or an Alzheimer's study, the funding can come from a lot of different places. As I said before, the researchers are in charge of applying for those grants, but it's not just limited to government agencies like the USDA or the FDA or APHIS for, you know, specifically. It also has to do with things like the military. For fracture type studies, the military would definitely fund something that has to do with healing wounds faster, for example. But also the private sector. A lot of agriculture studies are funded directly by industry as opposed to the government. A lot of food studies.
Calcium uptake.
Definitely.
All kinds of, you know, different studies. Rats are really important on. We have metabolic rats that we can take their feces and urine and see what, you know, run studies, run tests on them, so we can see all that. But just research in general is so important for the lives of so many people that we are proud to be part of this, and we appreciate you looking into our little world here of lab research and the care and love and compassion that we have for our animals is unbelievable. I mean, we care for these animals more than, sadly, a lot of the children in the world now are cared for. And so they never have a hot day, they never have a cold day, they never have lack of light, you know, whatever, you know, however we can make their bodies and their happiness the best, because you're not gonna have good research if you do not have good husbandry. And thanks to people like Alex and other people that are our animal caretakers, we really appreciate all the work that our caretakers put into this, and the dedication. It's really truly a love. So if you ever find yourself, you want to have a job, or as a student, all the way up as a full-time person, up to the lab animal vets, look us up, and we'll be glad to answer any questions. Thanks so much, Alex, for speaking today.
Oh, definitely.
I appreciate it.
Yeah, so thank you guys for listening to
Yeah.
all this information.